Hello everyone, welcome to JK Biology. In this video, we'll see the anterior, posterior axis formation in Drosophila. Anterior, posterior axis formation in Drosophila. First of all, we should know the basic concept, that is which one is anterior region and which one is posterior region. Anterior region, that is head region. Head region is called as the anterior region, and posterior region, that is abdominal end region. Abdominal end region is called as the posterior region. Is it clear about the anterior region and posterior region? And this anterior posterior axis formation, anterior posterior axis formation occurs before fertilization. Recognition of the anterior posterior axis occurs before fertilization. And for this anterior posterior axis formation, there are genes are involved. With the help of these some genes, there will occur the anterior posterior axis formation, and these genes are called as the maternal genes. The genes which help for the anterior posterior axis formation are maternal genes or maternal effect genes. Maternal effect genes. Now, why the name maternal genes or maternal effect genes? Because these genes are placed in oocyte. The genes are placed in oocyte by mother fly itself. By mother fly itself. And these are transcribed from, the genes are transcribed from mother's DNA. Mother's DNA. Hence the name maternal genes or maternal effect genes. Now next we'll see the list of maternal effect genes. Maternal effect genes. There are many maternal genes are there. Those are 1. Bicoid, second nanos, third one hunchback, fourth one caudal, fifth one Stephen, and sixth one Oscar. These are the maternal effect genes or maternal genes. Next we'll see the structure of egg chamber. Here we'll draw diagram of egg chamber. These are the nerve cells. And it is a oocyte. And nerve cells and oocytes are covered by a layer of follicle cells. Those are ovarian follicle cells. Here this is the layer of follicle cells. Okay, these are the anterior follicle cells. And these are posterior follicle cells. Here is a oocyte. And these all are nerve cells. And this structure, this is bicoid mRNA. This red color portion, this is the bicoid mRNA. Bicoid mRNA. And these regions, these zigzag regions are nothing but nanos mRNA. This is the nanos mRNA. This is nanos mRNA. This is our the egg chamber. Next, we'll see the function of these nerve cells. Nerve cells, these nerve cells produce mRNAs. What is the function of these nerve cells? Nerve cells produce mRNAs. Which are required for the anterior-posterior axis formation. Those are, first one, bicoid mRNA, nanos mRNA, hunchback mRNA, caudal mRNA. These mRNAs are produced by the nerve cells and they are transported inside the oocyte. And after fertilization, this is mRNAs are produced before fertilization, and after fertilization, see what happens after fertilization. These mRNAs are translated into proteins. mRNAs are translated into proteins. Now, inside the oocyte, there is a presence of the proteins. Those are bicoid protein, nanos protein, hunchback protein, caudal protein. These are the proteins which are present inside the oocyte after the fertilization. Now, among these proteins, some are required or some are helps in the formation of the anterior axis and some of them help for the formation of the posterior axis. Now, we'll see the which proteins are required for the anterior axis formation or what are the reason for the anterior axis formation. This anterior axis is formed due to presence of two proteins. Those are, one is bicoid protein and hunchback, and posterior axis is due to presence of two proteins, that is, one is nanose protein and caudal protein. This is very important. You have to remember anterior axis is formed due to presence of the bicoid and hunchback protein. Posterior axis is formed due to nanose and caudal protein. Next, we'll see the concentration gradient in oocyte. Here, concentration gradient in oocyte. There are two graphs are there. One, this graph is, first graph is before fertilization. Second graph after fertilization. Now, see the first graph. Before fertilization, in the oocyte, there will be presence of the mRNA molecules. Now, in the anterior portion, before fertilization, the mRNA, which mRNA is more in concentration, that is, bicoid mRNA is more in anterior region, and there is no presence of the nanose mRNA. But in the posterior region or posterior portion, there is a high concentration of the nanose mRNA, but there is an absence of the bicoid mRNA. But before fertilization, both hunchback and caudal mRNAs are present equally throughout the oocyte. And here, after fertilization, second graph which shows, see here, after fertilization in the anterior region, there is a high concentration of the hunchback and bicoid and less concentration of the caudal and nanose. And in the posterior region, there is a high concentration of this nanose and caudal and less concentration of the bicoid and hunchback. This is the concentration gradient after and before fertilization. Is it clear? Yes. We'll take down that before fertilization and after fertilization. Now, before fertilization in the anterior region, anterior end or anterior region, high concentration of bicoid and no nanose protein. And in the posterior end, high concentration of nanos protein and no bicoid. Sorry, not protein, it's a mRNA. Before fertilization, there will be presence of the mRNA, not the protein. Bicoid. mRNA. Nanos mRNA, and here no bicoid mRNA. Is it clear? This is before fertilization. And other two mRNAs, hunchback and caudal mRNA. These are equally distributed throughout oocyte. This is before fertilization. Remember, before fertilization in the anterior end, high concentration of the bicoid mRNA and there is no presence of the nanos mRNA. In the posterior end, there is a high concentration of the nanos mRNA and there is no presence of the bicoid mRNA. And hunchback and caudal mRNAs are equally distributed throughout the oocyte. Next, we'll see the after fertilization. Next one, after fertilization. In the anterior region, high concentration of bicoid and hunchback and very less concentration of nanos and caudal. Next, in the posterior region, very high concentration of nanos and caudal protein and very less concentration of bicoid and hunchback protein. Is this clear? This is about the concentration gradients in oocyte, that is before fertilization and after fertilization. There are some other two maternal genes are there. Those are, one is Stephen, another one is the Oscar. And these two genes are involved in the translation of nanos mRNA. These two Stephen and Oscar genes are involved in the translation of nanos mRNA. See how they are involved in the translation of the nanos mRNA. Nanos mRNA, it is coded by two proteins. Those two proteins are, one is smug protein, smug and cup protein. In the initial stage, the nanos mRNA is having the two proteins. Those are the one is smug protein, another one is the cup protein, and these two proteins will act as the translation inhibitors of nanos mRNA. These are inhibitors of these two genes, smug and cup proteins. These are translation inhibitors of nanos mRNA. That means these two proteins, presence of these two proteins will not allow the translation of the nanos mRNA. For the translation of the nanos mRNA, these two proteins should be removed from the nanos mRNA. Now, this Stephen protein will help for the translation of Oscar mRNA. Stephen protein will help for the translation of Oscar mRNA. After the translation, Oscar mRNA will form the Oscar protein, and later this Oscar protein will remove the two proteins, that is smug and cup protein. Once after the removal of these two, smug and cup protein, from the nanos mRNA, it will lead to the translation of nanos mRNA. Is it clear? That is Stephen and Oscar mRNA, which helpful for the translation of the nanos mRNA. This is about the anterior-posterior axis formation in Drosophila. If you like my video, please like, subscribe and share. Thank you.